Hello, everybody, and welcome. Today we're going to be talking about how DNA replicates. For your assignment, I would like you to answer these questions, which will be available for you on a Google form. So you can pay attention to the slideshow as I'm going through and kind of look for the answers to these questions. I know you're going to have to draw and label the nucleotide in your notebook and show me when you're back in person next week. So there's some major concepts here. These people all had big parts in the development of our understanding of how DNA replicates. These are your keys to this whole thing, so hopefully you pick this out as we go through it. Just kind of a little bit of science humor on a Friday morning. If you don't understand it yet, you probably will by the time we're done. So what we're going to come out of this, what we're going to get out of this, our objectives are here on this slide. You should understand the structure of DNA. You should understand how we came to that understanding of the structure. You should come to the understanding that how replication occurs and that it's what we call semi-conservative. You should understand what that means by the end of this. Then there are some major enzymes that you need to kind of pin down here. DNA polymerase is a big one. Ligase, helicase, topoisomerase, all very important. Also understand the difference between bacterial and eukaryotic chromosomes and how they replicate, and then kind of how gene expression can be affected by the way DNA is kind of put together in a cell. So the big problem is, or early on was, we didn't understand as a science whether DNA or proteins were the genetic material within organisms that was being passed on from generation to generation. So some experiments needed to be done to get to that understanding. One of the first people to kind of try to tackle this was a man named Frederick Griffith, and he experimented with different types of cells that would, when given to mice or humans, it's not really ethical to work on humans. It's not terribly ethical to work on mice this way, honestly, but that's beside the point. At this point, this was done in 1928. Can't really get too mad at the guy now. He's dead. If you take S cells from this strain of bacteria and inject them into the mouse would die of pneumonia. R cells of the same type of bacteria could be given to these mice and they would not die. The difference between S and R, if you look at the picture here, is the S strain has this smooth coat around the outside of the cells, whereas the R strain, which is considered the rough strain, does not have the coat. When you heat kill the S cells and inject that into the mice stay alive. But if you take R cells and mix them with heat-killed S cells, inject that into the mice die, and then from their lungs you can isolate living S-type cells. So what this is saying is that there's something being transferred from the heat-killed S-type cells to the living R-type cells, and Griffith was one of the first people to try to understand the system. What was happening here was that transformation was happening. Now, we didn't know what transformation was at the time, but Griffith kind of gave us the term transformation because the R-type bacteria were not harmful, but could be transformed into the deadly S-type bacteria by taking something from the heat-killed S-type that was mixed with them. So then along came these people, Avery, McCarty, and McLeod in 1944, and they tested the different candidates for genetic material, DNA, RNA, and proteins, and what they discovered was that it was DNA that was being taken in by the bacteria that was allowing them to become pathogenic. Hershey-Chase in 1952, so you can see that this is taking a long time to kind of come to this understanding, and part of that is because our technology at the time wasn't great. But Hershey and Chase worked with a small virus called a bacteriophage. It's a virus that affects bacteria. It's composed of DNA and protein, and what they did was they took the protein that the bacteriophage uses to make its coat its head and its outer shell of the virus, and they gave it radioactively labeled sulfur. That was a part of that coat of the virus. They also gave it radioactively labeled phosphorus. Then they allowed them to infect bacteria. And so they gave them the sulfur, allowed them to infect the bacteria, went through this whole process. And what they found was that after going through with batch one here, the sulfur ones, they didn't find any of the radioactive material inside the bacteria, which means the protein that they had radio labeled was not being injected into the host cells. The phosphorus radio labeled DNA, however, was found inside the newly infected bacteria, which means it's DNA that is being used to create the infection and to reproduce the viral particles inside the bacterial cells. So the conclusion is DNA is our genetic material. It was actually a really elegant and well thought out experiment, and it answered a very age-old question. So then came the questions about how is DNA actually built? And this answer took a long time also to kind of get to. Ultimately it came to Maurice Wilkins, Rosalind Franklin, and then Watson and Crick, who, they kind of get the credit for everything, but we'll talk about that in a second. The first guy that really started to understand how DNA was put together was this guy, Edwin Chargaff. In 1947, he published a paper that held that DNA composition, species to species, was different because he was looking at the ratios of adenine, thymine, cytosine, and guanine among the different species. And you can see a data table there down at the bottom from salmon sperm that he looked at. And what he discovered was that the percentages of adenine and thymine were always roughly equal, and the percentages of cytosine and guanine were also always roughly equal. So you can see that right here in the middle column. Right in here, he actually gave us the understanding, or the beginning of the understanding, that adenine and thymine are always paired together in DNA, and cytosine and guanine are always paired together. Then along came Rosalind Franklin. This is her right here. And for those of you who are in class, have watched the video about photo 51. That is photo 51 there, in section in B. Photo 51 is famous because it is the photo that Watson and Crick used to finally understand the actual structure of DNA. They were able to use this to determine that it was a double-stranded helix, because initially they thought it might be a triple-stranded helix. But because of Franklin's work, this structure of DNA was discovered. Now, unfortunately, she didn't get much credit for it, and they were able to publish their results. But by the time they published, she had already died, and so was unable to get much credit for what happened and what they discovered. And it's a pretty unfortunate piece of history, I think. She worked with Maurice Wilkins. They did this process called X-ray crystallography, where they would spend hundreds of hours exposing X-rays, like the one you see down here in the bottom of the screen, of different biological molecules, and DNA happened to be one of the ones that they worked on. And they also were able to provide measurements to determine how many nucleotides there were per nanometer of DNA, which was actually pretty interesting. Watson and Crick in 1953 had gotten a hold of Ros Franklin's picture and used this because they were in a race with Linus Pauling, kind of an unofficial race, with Linus Pauling to publish the structure of DNA. And because of Rosalind Franklin's picture, they were able to understand that the nucleotides were actually on the inside of the helix and not on the outside of the helix. And as a result of that, they were able to come up with the first working model of DNA. So we now understand that DNA has a backbone, a sugar phosphate backbone that is a repeating motif. So you can see here there is a phosphate connected to a sugar. That's the deoxyribose sugar connected to a phosphate, connected to a sugar, and you just continue that ad nauseum through the whole thing. And then on the inside of the helix, where the rungs of the ladder are, you find the nitrogen bases, thymine, adenine, cytosine, and guanine. The nitrogen bases themselves, adenine, guanine, thymine, cytosine, tend to be broken up into either purines or pyrimidines. Adenine always pairs with thymine, cytosine always pairs with guanine. Adenine and guanine are what we call purines. If you look at them, they have 2 rings in their structure. And thymine and cytosine are the pyrimidines, single rings in their structure. And you can also see from this picture here that adenine and thymine only form 2 hydrogen bonds per ring or per base, where guanine and cytosine form 3. And that's the biggest difference between them. So these hydrogen bonds hold the molecule together like a zipper. And they're not individually strong, but when you have literally millions of them together, they can become relatively strong. And they're good at holding the DNA together. You can see here the distance between the spirals of the DNA. This would be the major, the large outside edge of the spiral, and this is the minor groove over here. So you can see that the major groove is about 3.4 nanometers across, and that's not very far, obviously. The strands are anti-parallel, which means they run in opposite directions to one another, and that's important, as you'll see in a minute. But DNA has what we call a 5' end and a 3' end. That is just kind of, if you look at how it's set up, the 5' end here has the phosphate at the end of it, and the 3' end has just an open hydroxyl group. But they run in opposite directions to each other, and that's really important in the way DNA replicates, as you'll see. DNA is always packaged up because, honestly, you have so much of it in every cell that you need a way to coil it up, a supercoil, into these nucleosomes, which you see here in this picture, wrapped around histone proteins. And what that does is it allows the DNA to be packaged down into an incredibly small package so that it can fit inside the nucleus of a cell to be used when cell replication or protein synthesis is necessary. So ultimately, the only time you really see the chromosomes is when they're preparing for cell division. At this point, they will form up into chromosomes. Most of the time, DNA is in a loose form called chromatin. Chromatin itself, I'll move my picture here. Chromatin itself is this loose granular structure inside the nucleus until it's time to start forming up the chromatids, which are these 2 rod-like structures of DNA, that 2 of them form a chromosome. This isn't going to be seen unless the chromosome is, or unless the cell is getting ready for or in cell division already. So to compare prokaryotic and eukaryotic DNA, we need to kind of look at a couple of things. There are some similarities, there are some differences, okay? So when we're looking at, I'll move my picture again. When we're looking at prokaryotic DNA, yes, it's still double-stranded, all DNA is. It is a circular molecule, unlike eukaryotic DNA, which is linear. Bacteria tend to have just 1 chromosome, although you can also see in them plasmids, which is an extra chromosomal piece of DNA that is not considered part of their genomic DNA. The 1 chromosome they have, the ring that they have, is found in the cytoplasm. It is supercoiled into what we call a nucleoid, but they don't have histone proteins. Eukaryotic DNA, again, is double-stranded. It's linear. There's usually more than 1 chromosome. Usually, I say, but that varies from species to species. It's found in the nucleus, and it's often kept, most of the time, kept in the form of chromatin, as I said before. It's DNA wrapped around those histone proteins, and it looks granular if you were looking at it through a microscope. So the big question is, how does DNA replicate? And it was a kind of a difficult question to answer because there were 3 models of DNA replication. The conservative model, which said that one parent DNA strand would just replicate, and you still have that parent DNA strand all the way down through the generation of all the replicated strands of DNA. The semi-conservative model says that the parent strand separates into 2 individual strands, and then each of those go on into the offspring strands of DNA. And then you have the dispersive model, which says that pieces of the DNA are found in each of the 2 new daughter strands that are formed. So Meselson and Stahl were the ones that kind of answered this question by radio labeling DNA in their bacterial cultures. They gave them these radio labeled DNA strands, and then they let them reproduce. And what they found afterwards was that all of the offspring bacteria had gotten DNA that had one of the original strands in the first generation after reproduction, which means if you have one strand in each of the 2 replicated chromosomes, or not chromosomes, double strands, that means you're doing semi-conservative, but they had to do a second replication to make sure that it wasn't the dispersive model, which would have, all the strands would have had a little bit of DNA with radio labeling. By doing it this way, what they found going through the second replication was that only 2 of the 4 strands in the second replication had the originally radio labeled DNA. So they identified the model as the semi-conservative model, and that's the one we go with. We are pretty well aware of how it works now. The awesome thing about DNA is that it's actually self-replicating. All it has to really do is unzip, and then some work, the polymerases and the other enzymes come in and they can assemble the DNA according to the template found on each separated parent strand of DNA. So here's a replication video that you guys can watch. I'm going to take a break right now because school's about to start, so I got to get ready for first period. So I'll publish the second part of this at a later time, and I will see you guys next week.